So our next speaker will be Evangelos, and Evangelos is a faculty member at the Les Turner Center, and he recently received a very generous award from the Stem Cell Institute. Is that right? Maybe you can tell them the correct name, and I would like to congratulate him. Thank you.
Hi, good morning, everybody. Can you hear me well at the back? Yep. So, good morning, everyone. Thank you all for being here. Thank you to Hande for organizing this symposium. I'm always excited to participate. This is the 5th one that I'm here, and I feel every year that the winter comes earlier in Chicago. So, we heard a wonderful introduction about ALS from Uday, and what I want to do over the next 20 minutes or so is give you an update on a project that we've been working intensely in the lab over the last couple of years, in trying to understand how a very unique mutation in this gene called C9ORF72 leads to neural dysfunction and eventual degeneration in ALS. But as we heard from Uday, ALS is a genetically very heterogeneous disease, and the overwhelming majority of the disease is of a sporadic nature, and there's all these genetic cases. Now, what we are fascinated about is the fact that the genes that are known to cause ALS encode proteins that seemingly do very, very different things. And some people have suggested that you can subclassify them to distinct functional categories. And what the overarching question that we're interested in addressing is trying to understand whether there is any molecular unification amongst the different genetic subtypes of ALS, and whether this has any validity or any broader relevance in the sporadic disease. So the approach we're taking to address that is we're creating patient-specific models for a number of these genetic subclasses, and we're studying them individually. And things that we discover to be relevant in one subtype, we then try and see whether they're relevant in another. And we have projects in the lab looking at C9ORF72, SOD1, and some of these other newer mutations. But today, I'd like to tell you about some work that was spearheaded by Alberto Ortega, a postdoctoral fellow who's sitting there in the audience, and Beth Daly on C9ORF72. So this is a very unique mutation. Unlike the other ALS causal mutations, this is a repeat expansion. This is the hexanucleotide sequence. It's 4 Gs followed by 2 Cs. And normal individuals usually have about 30 or less of these repeats. This is an intron of a gene which encodes a protein which is likely involved in endosomal trafficking. Now, in ALS cases, these repeat expansions expand, and patients are known to carry hundreds, in some cases thousands of these repeats. So we know that the disease can be, there have been suggestions that the disease can be mediated through a combination of loss of function as well as gain of function effects. We know that the expression of the mutant allele is downregulated in patients. And we also know that RNA is transcribed from these repeat expansion, and RNA is translated through non-canonical ways to create these 5 dipeptide proteins. And work over the last few years from a number of different labs has shown that both the RNA and the dipeptides can be toxic through gain of function effects. Now, these are the dipeptides here, and there's 3 dipeptides in the sense strand and 3 dipeptides in the anti-sense strand, but one of them is the same, so there's 5 dipeptides in total. So I'm summarizing a lot of work from a lot of different groups over the last few years, including some work from Chris Donnelly's lab. But what we know about these toxic gain of function effects can be summarized in this slide. So we know that the RNA binds and sequesters RNA binding proteins in RNA foci that are predominantly nuclear. This is shown here. And we also know that these dipeptide repeat proteins bind and sequester a number of proteins that have these so-called low complexity domains, and they can form aggregates which are predominantly cytosolic. Now, what we also know is that components that are implicated in trafficking between the nucleus and the cytosol, nucleocytoplasmic trafficking, are strong genetic modifiers of these gain-of-function toxicity disease in various models. So we reason that if this model is true, what should happen in C9 ALS systems is an imbalance between the nuclear and the cytosolic proteome relative to what happens in control cells. So we sought out to address this really simple question, which is which proteins are altered in their nucleocytoplasmic distribution in cells that express these repeats, and do these contribute to toxicity and dysfunction and eventual toxicity. Now, to do this, we initially wanted to set up a sensitized system. And to do this, we got these plasmids from Paul Taylor's lab, which include either a control number of repeats, 8 repeats shown here, or a high number of repeats, 58 repeats in this case. These are tagged with GFP, so when we transfect them in hex cells, we can see the green cells, we can fax purify the green cells, and then we performed a precise biochemical subfractionation to isolate either the nuclei or the cytosolic components of these cells. Now, we followed this up by doing mass spec analysis to identify proteins that were found in the cytosolic or the nuclear fractions. And this is just some fax plots down here and the pictures of the cells. Now, at the end of this, we had abundance values. These are normalized spectral abundance values for individual proteins. And because we had a fraction in the nucleus and a fraction in the cytosol, by doing this simple calculation, we could investigate what proportion of the protein was found in the nucleus versus in the total cell, and what proportion of the protein was found in the cytosol. Now, to validate our approach, we initially focused on known cytosolic markers or known nuclear markers. And if you look at the cytosolic ratio of cytosolic markers, it's close to 1, whereas the cytosolic ratio of nuclear markers is lower than 0.5, and in some cases very, very low. And this is just a visual representation of one of those markers in the cytosol. These are 3 samples, control, and 58 repeats, cytosolic and nuclear fraction. And you can see that the cytosolic marker is always in the cytosolic fraction, and the nuclear marker is always in the nuclear fraction. So at the end of this simple experiment, we identified about 10,000 individual proteins in all 3 conditions with no statistically significant differences in the number of proteins amongst the different conditions. But when we looked at the average cytosolic ratio of the entire proteome, so the average cytosolic ratio of these 10,000 proteins, we found a tiny but highly significant shift towards a slightly more cytosolic proteome. This suggested that proteins shift from the nucleus to the cytosol to some degree. So this is represented here. This is done after, this is a volcano plot. On this axis, we're looking at the shift. So proteins that are here become enriched in the cytosolic fraction, whereas proteins that are here become enriched in the nuclear fraction. And this is the significance scale here. So I hope what you can appreciate here is that this axis seems to be, there's more weight to this on the right hand, which basically is telling us that there's more nuclear proteins that shift towards the cytosol, about 56%, but however, there are cytosolic proteins which tend to shift a little bit more to the nucleus. Now, importantly, there were only 126 proteins, which correspond to about 1% of the protein that were identified, that passed the significance test. So this shift is driven by a small but highly specific group of proteins. So these proteins seem to be functionally related, and when we perform gene ontology analysis, there's essentially 3 pathways that become enriched: RNA metabolism, protein synthesis and translation, and cell cycle. I want to remind you that we did this in hex cells, so we're not particularly excited about cell cycle because we're interested in what happens in humans. But we're very excited about RNA metabolism, protein translation. Now, we've done a lot of validation on some of these proteins, but the one that we got really excited about was one of the proteins that showed some of the difference of the highest magnitude, which is a protein called ETF1, which stands for eukaryotic termination factor protein 1. And this protein is involved in both of these pathways, RNA metabolism and protein translation. What it does is it identifies a stop codon during protein synthesis, and it brings in a complex to cleave off the elongating peptide at the end of protein translation. Now, in cases where translation stalls because there's a premature stop codon or an irregularly shaped RNA, or just to regulate protein transcriptional level, instead of translating this protein, it brings in another protein called UPF1, which becomes phosphorylated and degrades this RNA instead of allowing it to make protein. This is a very well-conserved mRNA surveillance pathway known as nonsense-mediated decay. So ETF1 is typically a cytosolic protein, which we found became a little bit more nuclear. So we wanted to really understand why does this happen. But first, we wanted to figure out whether this was relevant in a more physiological cellular system. So for this, we turned into iPSC models. And this is just a representative Western blot showing you that indeed in C9 motor neurons derived from C9 patient lines, ETF1 becomes a little bit more nuclear relative to controls. And when we did careful immunocytochemistry to try and really understand where this happens, we noticed that this is a three-dimensional reconstruction of the entire nucleus of a stem cell-derived control neuron on the top, and a stem cell-derived ALS neuron on the bottom. The blue is DAPI, and these red spots bind C9ORF72, indicate ETF1. And we found that ETF1 accumulates in this distinct nuclear foci. So this is just the quantification of this data. This is neurons derived from 3 C9a ALS patients relative to 3 unrelated healthy controls. And I hope you can appreciate that when you look at the nucleocytoplasmic ratio, ETF1 becomes a little bit more nuclear in the patient neurons. So why does this happen? Why does ETF1 become a little bit more nuclear? So we looked at we tested a number of different hypotheses, but to cut a long story short, what we eventually found was that ETF1 seems to accumulate in this discrete puncta that I showed you earlier, seem to coincide or accumulate in a very elaborate network of lamin B1 inner nuclear envelope invaginations. This is serial confocal imaging going from the top to the bottom of the cell. And we see this stunning architecture of the inner nuclear envelope in both the control and the patient motor neurons. But significantly, the patient motor neurons seem to accumulate a lot more of these protein in this puncta. Now, to get it, we got intrigued. Reviewers also got intrigued by this, and they asked us to take a closer look at it. So to do this, we performed expansion microscopy. This is a very clever technique devised by Ed Boyden a few years ago. But what this allows you to do is essentially blow up the nucleus by a chemical formulation. This is the same magnification. And when Beth tried this, she basically blew up this nucleus to this size. And when she then performed confocal imaging, we're able to take a closer look at these structures. And they're really fascinating because, first of all, they're like cytosolic intrusions into the nucleus. I hope you can appreciate that these structures here are DAPI negative. This is the inner nuclear envelope here. And I hope you can see that ETF1, our favorite protein, seems to accumulate deep within these structures. Now, importantly, we see these in control and patient neurons, but it is what's inside these structures that distinguishes the patient and the control neurons. And ETF1 is just one of those proteins that seems to accumulate in there, along with other proteins that are involved in nonsense-mediated decay and so on. So we also wanted to take a look at these in live cells. So for this, we used a lamin B1 GFP knock-in reporter line. This is live cell imaging. This is confocal imaging. This is a healthy neuron, about 30 days old. This is a 20-minute movie. And I hope what you can appreciate is that these structures are highly dynamic. So they seem to come in and out of focus. They seem to appear and disappear. So we think that these represent a functional component of motor neuron biology. We're really fascinated and want to do further work trying to understand what it is that they do. But what we want to focus for the rest 5 minutes is what we know, what the role of ETF1 and the accumulation of ETF1 within these structures in motor neurons. So I told you that ETF1 is implicated in 2 processes, protein translation and nonsense-mediated decay degradation. So we looked at these processes, and we found that in C9 patient motor neurons, de novo protein synthesis, so translation seems to be decreased. This decrease was done, was ETF1 dependent. In experiments I'm not showing you, we showed that when you remove ETF1, you basically block translation in controls, but there's very little effect in the C9 cells. And in contrast to a reduction in translation, we see an induction of markers of nonsense-mediated decay. This is one of those markers called phospho-UPF1, normalized to total UPF1. This is the spread of the intensity of phospho-UPF1 in control neurons. And when you see, when you examine C9 patient neurons, you see that there's a shift towards neurons showing higher levels of activation, of higher levels of phospho-UPF1 to be more specific, which likely suggests higher NMD activity in these cells. So what I've told you is that ETF1 seems to accumulate in these invaginations, these cytosolic intrusions deep into the nucleus in C9 patient cells, and at the same time, protein translation is reduced, and mRNA degradation by NMD seems to be induced. So it dawned to us when we looked at this data whether that one possibility is that ETF1 might be targeting the expanded C9ORF72 transcript itself for degradation through this nonsense-mediated decay pathway, because it is a prime candidate to be degraded through NMD, because there's an intron that's retained, it has this irregular large shape, and so on. So to begin to address this, we did a couple of different experiments. I'm just going to show you one in the interest of time. These are the C9 RNA foci that I told you are typically found in C9 motor neurons, and they're in most cases they're nuclear. But when we knocked down ETF1, or removed ETF1 from patient neurons, they disappear from the nucleus and they started accumulating in the cytosol. This was quite a dramatic difference, which is quantified here. And neurons derived from 3 independent C9 lines. This is the reduction in the nuclear abundance of the foci and a concomitant increase in the abundance of the cytosolic foci. So we think that this suggests that the expanded C9 transcript itself is targeted by ETF1 for NMD-dependent RNA degradation. And this is just the model that we're putting together. So motor neurons that have C9 mutations accumulate this ETF1 within the structures. They reduce protein translation, they induce NMD-dependent degradation, and they also degrade some of the C9 transcript. Now, to validate this model in an in vivo intact nervous system, we collaborated with Uday Pandey's lab, and we went to these beautiful fly models so that Uday has developed. So these flies either express 30 or 36 repeats of C9ORF72 specifically in their eyes. And what you can do with this, as Uday nicely explained, you can manipulate your gene of interest, either overexpress it and knock it down, and by reading off the effects on the toxicity on the fly eye, you can understand whether there's a genetic interaction. So when we did this experiment, we found that, as we would expect, when we knock down ETF1 in the fly, the phenotype becomes worse, whereas when we overexpress ETF1 in the fly eye, the phenotype becomes suppressed. This is just quantified here, so this is the RNAi knockdown and the ETF1 overexpression. Now, if our model was correct, the mechanism by which ETF1 would affect toxicity would have to implicate DPR production, because I showed you that ETF1 blocks the exit of the expanded transcript from the nucleus and its downstream translation into DPRs. So we did some dot blot assays in collaboration with Uday and Sucleen, who's also sitting in the audience. And I hope you can appreciate it, so that when we knock down ETF1 in these flies, there's more production of this poly-GR dipeptide, which is one of the most toxic dipeptides in this model, whereas when we overexpress ETF1, the production of this DPR seems to be suppressed. So that's it. That's all I wanted to tell you. Again, I'd like to acknowledge the people that did this work. So Alberto Ortega is right there, and Beth. So they're a super team working on the C9ORF72 story for the last couple of years, and we hope that this will be published soon. And I'd also like to acknowledge all the other people in the lab that work on distinct ALS projects, and hopefully I'll be telling you more about those in the future. Big thank you to the Les Turner ALS Foundation for supporting this work, the NIH, and also I'd like to acknowledge the New York Stem Cell Foundation, who will be supporting my lab over the next 5 years. So thank you for your time, and I'll happy to take any questions.
So we have time for questions. Maybe I can ask the first, if you don't mind.
Yeah.
You talked about those stress granules, and there's a big debate in the field that the stress granules can actually be helpful, for cells to maintain their homeostasis and health, or that the stress granules, when you have them, it's a sign that these neurons are going to degenerate. Can you tell a little bit about the role of stress granules in motor neuron degeneration?
Yeah, so this is something that we haven't touched at all. We haven't looked at stress granule formation in our C9 models or any of our models. We think we'll have to address that point, for some of the other projects that we're doing. Perhaps this is Chris Donnelly, who's sitting in the audience, is probably the best person to address this question. I know has an interest in this debate on stress granules.
Any other questions? No questions?
Yes, Bob.
That was really incredible. Those moving in cytoplasmic invaginations, is that seen in other models beside this ALS model? We see it in other neurodegenerative diseases. Is it something very unique about motor neurons in ALS? Is there a general principle?
Yeah, so in short, we don't know. There's very little in the literature about these so-called nuclear invaginations. People have described these in fast dividing cells like cancer cells and so on. I've never seen live cell imaging of these structures. I believe our data, as far as I know, is the first time anybody has looked at them. There is a recent report, actually, a recent paper that was published in Cellular Reports earlier this year, showing that tau, overexpression of tau, or mutant versions of tau, can induce similar structures in cortical neurons. As far as I know, that's the only mention of these structures in the central nervous system, but maybe somebody else has described them before. As to whether they're specific to motor neurons, I don't think so. We're one of the projects that we're undertaking is cataloging the incidence of this and how much space they take, their structure in distinct neuronal subtypes, both in vivo with stem cells, but also in vivo in controlled tissue. In data that I didn't have time to show you, we see these structures in postmortem cortical tissue as well. And the spinal cord things seem to be a little bit more complicated, but we're really interested in figuring more about these structures.